got a slide deck here for you about this study that we kicked off this last year, looking at our soybean inputs, our pest control inputs, and when they pay off, and what those scenarios are. A little bit of a misleading title because I won't talk about money today, really. Reason for that being, we've got 1 year of data. So we've got 6 sites a single year. One of the things we're most interested in with this project is how frequently these different inputs that we use pay off. But as an entomologist, I've got a number of other, kind of interests on the side. In particular, looking at what these insect populations are doing, when it makes sense to treat them, and which insects are really moving the needle in terms of yield. Trying to get a more uniform picture of this. One of the things especially that I'm interested in, we make threshold recommendations, right? We make recommendations on what's the economic threshold for bean leaf beetle, for Japanese beetle, for stink bugs, for stand reducers, for all these different insects. Since it's my job to develop those, I got a vested interest in making sure they're right. I want to make sure they're right. I want to put them to the test. And I want to see what happens when insects out in the field exceed these different thresholds. If I developed a single experiment and said, boy, I hope stink bugs are above our economic threshold in this experiment this year, that would not happen. In fact, if I put that trial out, I can pretty much guarantee it wouldn't happen. We can do some of that kind of work with caging studies. As you might imagine, when you cage insects on a plant, it causes some complications, right? One, your plot size is going to be small, like real small, if you do that in a cage. Two, insects, when you put them in a cage, they're a little bit like people when you put them in a cage. Some people respond okay to it. A lot of people don't. A lot of insects, you put them in a cage and they fly to the top of the cage and they look around for an excuse to die. That's what insects do a lot of times when you put them in these cages. Some take to it very well. Did my PhD on an insect called the kudzu bug that loved to be in a cage. We put them in cages and they just bred and fed and destroyed soybean plants. It was awesome. But not everybody's going to do that. And with the generally pretty sporadic, pretty unpredictable, kind of mixed complex of insect pests that we have in Illinois, and most places really, trying to get as many of these sorts of trials out there in as many different locations, and especially over a number of different years, is important in answering these sorts of questions. The other thing I'm going to point out to you here today, and I got my contact information up there on the first slide. I've got it again at the end. You don't hesitate to reach out to me if I can ever do anything for you. I've got probably more data slides in this talk than any talk that I've done in the last 5 years or so, probably since I was a PhD student. My intent is not to run through and read each one of these data slides off to you, because I know a lot of you and I like you, and I don't want to punish you. I don't want this to be an exercise in pain for anybody. But this gives us the material to talk from. I'd love to have a discussion. And if you've got some side questions, some side topics that we can run into with this, I'd love to go through and address those. So please don't hesitate to stop me at any time to talk through some of this stuff. And apologies to the folks online if that's hard for you, but I think it makes for a better discussion when we can do that. So the nature of our study, we're testing really some of the most common insect control tools that we use in Illinois. And a lot of our insecticides that we use, they're either applied to the seed or they go out at a growth stage, often with a fungicide, right? Often they're tank mixed with a fungicide, sometimes with a herbicide. I kind of resent that a little bit, so I wanted to look at just the insecticide. We want to get a look at just the insecticides. I don't actually resent that. It's just we want to isolate the effects of insects and insecticides. on this. We also want to get a look at some trials that we can really control everything. So we're doing this at our research farms. And at our research farms, we're doing a full complement of treatments. So we do plots with no insecticide at all. Whether or not it exceeds a threshold, whatever happens, we can leave those without an insecticide. If something came in and ate the entire plot, we can deal with that on our research farm, right? That's cool. That's what we have these farms to do. We'll have a seed treatment insecticide. All of this, I should point out, had a fungicide seed treatment on it. So we have the addition of an insecticide in treatment 2. And then we have an application at R3. We have an application at R5. And then we have all 3 of those things. So we have, if we had an insect out there, and we can kill it with the practices that we're typically using, we killed it. That's the idea anyway. We're also doing this on farm. We're doing this on commercial farmers. Couple of reasons I wanted to do that. One was because I like to get trials on farmers to make sure that what we're doing makes sense in the real world that y'all operate on, not just the university farm world that I operate on, right? The other reason for it is there are a lot of potential pest control inputs that we could use, right? I'm not going to be able to do all of them on our university farms. So this is an opportunity to get some different treatments in there to look at some things that are locally interesting, whether that's a different active, whether that's looking at some of the premix type products that have a neonicotinoid and a pyrethroid in them versus, we've been using basically Warrior. Lambda is the most common growth stage insecticide that goes out, so that's the one we use, but it's not the only one that everyone uses, so we want to get an opportunity to test some others. What I think separates this work from a lot of similar studies that I see is we are assessing the insect populations, and we're doing that pretty rigorously. So we're going out and sampling these fields every other week to see what's actually there, to see what sort of injury we're seeing in these plots, so that at the end of the year, if we see that our insecticide at R3 gave us 2 additional bushels, we have an idea of why that occurred. Very often, when you see treatments like this, that's the piece that's missing. Of course, the reason that's the piece that's missing is it's really hard to assess these insects in a rigorous way. That's where me and my gain of undergraduates and graduates come in. That's what we do. I'm interested in answering a number of questions, as I mentioned earlier, about how often these sorts of tactics pay off. Over time, we're hoping to build a pretty large data set with these so we can really answer a lot of questions about what happens in different years. What happens when it's a Japanese beetle year? Was this last year a Japanese beetle year? Not really. Certainly not around here. Everybody says it is somewhere every single year, but we didn't have much around here. What happens when we see it in a big bean leaf beetle year? What happens if, God forbid, soybean aphid comes back again and makes an appearance in Illinois? Hasn't done that in a few years. That's the reason we want to do this over time. So again, single year of data, right? We've got 1 year of data on these different trials. Year 1, we didn't exceed an economic threshold except in one individual plot at Monmouth. So we didn't exceed it for the treatment. We exceeded it in one plot. We'll talk a little bit about each of these individual trials as we go through. But in year 1, we didn't exceed economic thresholds. And generally speaking, I think that coincides pretty well with what most people saw in soybean last year, right? It was not a big insect pest year in soybean, certainly not around here, and generally speaking, I don't know, what we were talking about before the thing was cornleaf aphid. I think that was the most unusual thing we saw. Obviously, we didn't have cornleaf aphid in soybean. But I'm going to go through these one site at a time in some detail. And again, I'd like for you to stop and ask questions as we go. If nothing else, to make sure I don't get caught up in all these data slides and start just talking about F's and P's and the advantages of box plots that I used versus bar charts and that sort of thing. I'm sure you're all very interested in learning that. This was at South Farms. This was our location in Urbana. And these on-farm tests are plots we were looking at 40 foot by 150 foot generally for an individual plot. So not large from a field standpoint, but a lot larger than I often do on the university farms. So in this case, our insect pest pressure was generally pretty low, with one exception, which was bean leaf beetle. And as we've often done locally the last few years, towards the end of the season, we started to see these bean leaf beetle populations pick up a little bit. I don't know how many of you all have been seeing that in this area, especially on beans that are a little bit later. Those bean leaf beetles, they're mobile and they start to kind of funnel into the last beans that were green. These weren't particularly late, but we started to see those populations increase a little bit. And you can see a couple of interesting things here. One, when we first started to see these bean leaf beetles come up, and that was towards the end of August, we were still actually getting some reduction in our plots where we had an application out at R3. Which is interesting. That was, 16 days prior. So we're right kind of on the cusp of when that insecticide is still doing something. When we talk about a pyrethroid like that, 7 to 10 days is what we're generally thinking about. When we got in a couple weeks later, which was a little bit after the R5 application, and by the way, this first data point here, that was immediately before the R5 application. So we went out and sampled these, and then we sprayed them. That one, unfortunately, was sprayed with a backpack carried by me. It sucked. I don't want to do that again next year. We were able to use drones for a few of these, and that was a lot easier. I was not a happy camper on the 21st of August because of that. When we looked 16 days after that R5 application, we saw those populations rebound, where we had the seed treatment and where we had that R3 application, where previously we had knocked them back. But then you see where we had that R5 application, we kind of maintained them low the rest of the year. Again, that makes sense if you think about when these beetles were coming in and how long these materials last. And if you look at the kind of numbers we were looking there, we were up around 50 to 60 and 25 sweeps. Okay, a lot of you not using a sweep net. We take this net, it's about yay wide, 15 inches wide. We swing it through the canopy 25 times, and we count what we find. So if you've got 50 and 25 sweeps, that means every time you swing that net through the canopy, you're picking up 2 bean leaf beetles. So a lot. That's actually a pretty high population. It's not as high as some I'll show you when we talk about insecticide efficacy work, but it's quite a few bean leaf beetles. Our defoliation, though, remained, it remained below 3%. So we weren't hitting any kind of a economic threshold on defoliation. And when we looked at yield, we didn't see that come out in yield. So we had a lot of bean leaf beetles. We didn't have them at levels where we were impacting yield in that particular case. And in this case, on all of these trials, we sampled all the insects, okay? We sampled everything, pest, beneficial. We even counted some things like lightning bugs that, we tell them to count everything so that we don't miss anything, essentially. So we didn't affect yield in that case. Even though we had relatively high numbers in some cases of bean leaf beetles late, late in the season. That's what we're looking at first? Yes, sir.
Did you take into account between bean leaf beetles, stink bugs, and numerous other ones? It just seems like they prey on the pods and things there.
Yeah, we counted our stink bugs as well. So we counted everything, basically. And in terms of our analysis so far, kind of what I'm showing you is what we've got. We've got more to dig into with these trials, certainly, and especially as we start piecing together multiple site years. we can look at some questions like that, right? We can look at, okay, we've got subthreshold on bean leaf beetle, we've got subthreshold on stink bug. Do those two come together and actually cause a problem for us? Would controlling those pay off for us? We need to get some observations together to do that. But yeah, we're counting everything. We're counting everything that you can get in a net. We're doing stand reducers early. Didn't show that data because there's not much to show. It was a similar story. We're counting everything that we have the ability to count insect-wise out in these fields. We're also making observations on, you know, diseases and that sort of thing, if there's anything unusual going on in these trials. The idea is to get something pretty comprehensive by the end of it. Any other questions? Yeah.
How are you quantifying defoliation as a group?
Yeah, visual estimate. We're just doing a visual estimate. We're doing that at the canopy level right now. Oh yeah, sorry. I'm not repeating the questions. I already failed. I already broke the rules. So the question was, how are we quantifying defoliation? We're doing a simple visual estimate so far. We've got the ability to go out like with a phone app and actually measure the actual percent defoliation. We didn't get to levels where we felt like that was necessary this first year. Everything you're going to look at, the highest defoliation we recorded was below 5%. So nothing was heavily defoliated. We start to get to that 5% level, that's where quantifying it can kind of show some things. But yeah, we just did that with a visual estimate of the whole canopy, which is something we've done in a number of our trials over the years. Go to the next one. This was actually Monmouth. The most surprising result of this whole first year for me so far, Monmouth had the highest stink bug numbers of any site that we did. And if any of you who have worked with experienced stink bugs before, you know that generally speaking, the further south you go, the worse it is, right? As a general rule, all else being equal, you go to southern Illinois, we see more stink bug issues than we do up north. This year, this site, we saw quite a few of them at Monmouth. We also saw quite a few Japanese beetles. That's what you're looking at here. What was interesting here, we saw Japanese beetles before our R3 application went out. When we put that R3 application out there, they were already gone. So it's not that we killed them by spraying at R3, it's that they pieced out on their own and went and fed on corn silks or rose bushes or whatever, you know, whatever else, or they died. That happens to them sometimes too. And this is one of the reasons as we go through here, if you've heard me talk about this before, you know I'm generally speaking a little bit, you know, I would rather do a scout and spray approach than a growth stage based approach, generally speaking. That's generally what I would prefer to do for insect control. This is part of the reason. Part of the reason is that the period of activity of these foliar materials is not very long. And even when we do have insects show up at sort of a meaningful level, we often miss them with that kind of an application. But that's what we saw in terms of Japanese beetles at this site. So it happened to be, I think, about the highest Japanese beetle site that we had this last year as well. Stink bugs, we actually exceeded our threshold, which is 9 and 25 sweeps, okay? That's 1 per row sweep. That's, if you go out into this field, you are seeing stink bugs in the canopy. They're not hard to find at that level, if that's to give it something that's a little more relatable than a sweep net. When you have stink bugs at that economic threshold, and when we were walking through the plots at Monmouth in this trial, you're not having any trouble seeing stink bugs visually as you're walking through that field. You're seeing them all over the place. We're picking up between 5 and 10 and 25 sweeps in those untreated plots. Kind of interesting here, actually, we did suppress them both in the R3 and the R5, and of course, where we had both of those together, we suppressed them too, right? We can't kill what's already dead, but we managed to kill them in that as well. So we reduced stink bugs. We did not reduce Japanese beetles because they were reduced by nature before those applications went out. We didn't see any difference in yield at this particular site either. didn't affect yield by killing those, again, mostly sub-threshold, but close to threshold populations of stink bugs. That actually surprised me a little bit. I thought we were creeping up there with the stink bugs enough that we'd probably see something in those R3, R5 type applications where we suppressed stink bug numbers. But we didn't during this first year, which, okay, that's what we saw. So far, so good, right? Any questions about what we're doing here so far? Also, I failed to point out that all my data slides look exactly the same, so I apologize for that. That's because it's a lot easier to copy-paste my SAS code for producing a graph than it is to make a new one every single time. So I apologize that you're looking at the exact same color scheme for every single one.
So I have a question.
Yeah, what's up?
When you're spraying an R3, of course I use a ground rig. It's hard to not put on a lot of water just to get the penetration.
Mm-hmm.
And I mean, there's talk, I've been to meetings, you guys are talking about spraying still kind of R2, less foliage, getting better penetration coverage. I haven't done that. I'm basically only going to try it for some of it
Mm-hmm.
for this company. I didn't know if you've ever done anything early or not.
Gotcha. We didn't do anything, and the question was if we had done any of these applications before R3 in our research. We haven't. We haven't yet. That's something we could potentially look at on the foliar. I think that would probably be something that'd be easier to look at on the on-farm side. You know, to evaluate on-farm. One of the issues we run into with this experiment, so like when you're looking at these 5 treatments, okay, that's 20 plots. They're 20 relatively large plots. That's exactly the reason we're doing that on-farm work, is that we can look at some of these other treatments that don't necessarily fit in with these, and so that this experiment doesn't become unmanageable. And 5 treatments is about what we can handle for an experiment like this, given the stage, given the number of plots, and given the fact that we're going in here with nets every 2 weeks and swinging them through the canopy and counting what's there. But that is a good point, and that's something that, for instance, in this trial where we had those Japanese beetles early, yeah, I'm confident that if we had done a V6 application, it is about what that would have been. We were V6, V8 there. We would have seen a reduction in Japanese beetles where we sprayed them, right? And that's something, actually, for those of you who've looked at efficacy on Japanese beetles, like just can we kill it? Yeah, we can kill Japanese beetles with an insecticide. They're not hard to kill with a foliar pyrethroid. We're real good at that. We kill them when we spray them. Yeah, good question. 25. Okay, I'm doing okay. Sorry for peeking at my phone. It's to make sure I don't ramble, which is something the older that I get, the more I do. Next trial, we're over in Perry, Illinois, over in western Illinois at our research site there. Okay, this is an interesting one. It's in the river valley. It's a little bit different soils. You know, I haven't gone through and done anything with the soil type yet and that, but it's unique in terms of its characteristics compared to some of these other sites that we're looking at. And also, when you sit outside of these river valleys, sometimes you see some higher insect populations in that environment. We didn't see tremendously high insect populations this last year. We did have a little bit of elevation with Japanese beetle and with stink bugs. We didn't see enough of an elevation to get real clear treatment effects, like nothing that's going to bust out statistically on these. The other thing we saw that was kind of an interesting one for those of you who were in southern Illinois is Dectus stem borer. We saw quite a bit of Dectus stem borer actually at this site, which we didn't expect to see. We didn't do a very good job of killing them with the insecticides that we used, but we had them in there, and that's one that we could add into an analysis like this and see if we can find something that controls that insect. We had pretty good numbers on Japanese beetles, pretty good numbers on stink bugs. We saw a little bit of reduction in both of those, though not a tremendous amount as we got towards the end of the season. We saw a yield effect this year, but it was because we used a ground rig and we ran over our beans at R5. Anybody not know that that actually reduces yield? I actually knew that it did. We got stuck is why we had to do that. We got into a place where we had to use a ground rig there. We couldn't do what we originally planned. And sometimes it'd be like that, right? Sometimes that's what happens. We did not see differences in yield in those first 3 treatments. And maybe that's not terribly surprising considering where we were with our insect populations at this site. Ewing, finally. And this one, I don't know. I was optimistic we were going to get good insect pressure at this site down in southern Illinois. I always think, southern Illinois, we're going to get high stink bug numbers. We had pretty low insect numbers. We actually had the lowest insect numbers at this site out of all of them. We also had the lowest overall yield potential. That was less surprising, right? It's not very good soil we're working on there. It's a no-till farm. It was one of the first no-till demonstration sites, possibly in the world, certainly in the U.S., for a reason. But we did get some suppression there of, what are we looking at over there? Why can't I see it? Defoliation. This was the only site where our insecticides actually moved the needle on defoliation itself, on injury to that crop. So we're evaluating both the insect pests and the injury. And that's important, right? Because as you kind of mentioned, multiple different insects defoliate. So you could have these small populations of Japanese beetles, you could have these small populations of green cloverworm, small populations of beanleaf beetle coming in late. And in fact, when we see our thresholds exceeded for defoliating insects, that's usually how it happens. It's not usually just one. It would be real uncommon for Japanese beetles to come in by themselves and cause economic loss to a crop. It's when they roll in, cloverworm rolls in, beanleaf beetle rolls in, then you can get something. We actually saw a little bit of changes in green cloverworm at this site, and that was probably the insect most responsible for that defoliation we saw. Now, our thresholds for defoliation in soybean during the reproductive stages, they get as low as 10%. 10% of the canopy taken away. We lost upwards of 3% at this site. Didn't affect yield. Yield potential there wasn't very good. You'll note there's kind of a lot of variability, though the means are all just about the same. It's a low yield potential site. We did the best we could on that. But at those sub-threshold levels of defoliation, we didn't lose yield. Any questions here so far? Anything else that we can go into some additional detail about? I know it's a lot of data right after lunch. Did this in Vandalia down at a private farm. This is an example of the sort of on-farm work that we're trying to do. In this case, we had a field that was sprayed at R3, and we were looking at if an additional application at R5 would move the needle, would provide something for us. I don't have an insect slide here because we found literally nothing when we swept that field. We found one spotted stink bug and some southern corn rootworm, if you're familiar with that insect, not exactly a yield enemy in soybeans. Spotted cucumber beetle is the other name for it. We didn't find much in terms of insect pressure. We didn't move the needle in terms of yield, perhaps not surprisingly, with that additional application. Oglesby, we did this up at Illinois Valley Community College. If anybody's familiar with that, they've got a nice demonstration farm there. We did this with a drone. We did it with Dennis Bowman's spray drone that he just got. If we can do more of them like that in the future, that'd be the way to go. It's a hell of a lot easier than wearing a backpack and walking across these big fields and that. Not a lot of fun, y'all, if you've never done it before. This one, we also had some fairly high numbers of beanleaf beetles. Not as high as the Urbana site, but fairly high numbers. Perhaps not surprisingly, given it wasn't as high, we didn't move the needle on yield here either. Okay, 6 site years, right? We're doing this over time. The purpose of this is not to say don't use an insecticide. The point of this so far is to look at what are the scenarios where that insecticide is likely to pay off. the scenarios we've looked at so far, we're sub-threshold, or we're getting that application out after the insect pest had showed up, or really both. In all of these cases, we didn't move the needle on yield. That's what we've got so far. What we're really interested in learning going forward, okay, I don't know what an average field looks like, right? There's 10 plus million acres of soybean in Illinois in any given year. How often is this paying off for us? And especially, what are the scenarios that are most likely to make it pay off for us? What are the insect pests that are showing up for us that are really reducing that yield? What level are they hitting when it makes sense for us to do that? What's the timing? What's the timing? And one thing we find with insects, unfortunately, they don't behave for us, right? They don't come in at a predictable time. The insecticides that we use most of the time, it's not that there aren't insecticides that have residual activity, right? Like, we've got a few of them out there that'll last longer than 10 days. Tend not to be the ones we're using the most often, but we've got them. When do you put that material out there so that it's actually at a relevant rate when these insects are out there, and it's actually going to move the needle for you on yield? And so looking through data like this, again, this is why we always talk about scout and treat, scout and treat, scout and treat. With these growth stages, you're spinning the dice a little bit, right? You're spinning the wheel. You're hoping that insect pest is either there right now, or it's there within the next 7 to 10, maybe 14 days. How do we refine that a little bit? Or at least, how do we give you a good probability for, if you do an R3, how likely is it that you killed something? And that's ultimately what we're trying to do with these, right? We're trying to kill bugs. They're not products that create yield. They're products that protect yield. What do we need to kill and when to protect that yield? That's what we're trying to answer with this. If it's something you'd be interested in participating in, if you'd be interested in putting a trial out, let's do it. Give me an email or shoot the ISA personnel that you're working with an email. Let's do something. Oh, yeah, there's a sign-up sheet. Look at that. Look at that. It's fun going to meetings that I had zero role in organizing, because they're often better organized than the ones that I organize myself. That's a lie, actually. They're always better organized than the ones I organize myself. I don't organize extension meetings either. Someone else does that because I'm bad at it. Yeah.
So typically the industry was leaning towards, if you're going to spray a fungicide on soybeans, to have an insecticide in there based off of data that suggests that there's a synergism there.
Hmm?
Reasons for not including something like that within the trial to say, yes, we're seeing that synergism, no, we're not. Is it something you'd be looking at moving forward?
Yeah, and the question was, looking at insecticides mixed with fungicides and the potential for synergism there. I think that'd be a great question to look at in the on-farm work. I think that'd be a great thing to look at in the on-farm work. Again, the reason we didn't do it on the research farms, one, is because I'm trying to isolate the effects of insects, in a sense, in that work, to look at our insect thresholds. Two is because even the simplest treatment looking at that would double, if not more, the workload for us, and we can't do that and look at just the effect of insecticides, right? And it would be an interesting trial, and one of the reasons, probably the first reason that I thought of having the on-farm component, was to be able to look at questions like that, because that's the most common question we get. And let's be real, nobody's doing an R3 just an insecticide based on growth stage, right? Like, literally no one. They're doing an insecticide because it costs them $1.50 an acre, and they're driving across the field as, okay, we're going to throw an insecticide in there. There's been I've talked to some folks who feel like the insecticide in some cases acts as an adjuvant for the fungicide. That's kind of an interesting concept. Some of them are emulsifiable concentrates, like they could alter the characteristics a little bit. But yeah, that'd be a great question to look at in the on-farm type work. But the reason we're not looking at it in the research farm work there is just because then it's 40 plots instead of 20, and that reduces our ability to look at the question that we're originally trying to answer here on insects. Yeah, what's up?
The insecticides and treatments that are in theory combating rodents and pressure, most of your sampling is going to come from your hobbies. Why not try to go in earlier?
Yeah, that's a good point. Good clarification here. The question is, why weren't we sampling earlier to look at the effects of the seed treatment? And we actually were. So we actually were. We also sampled, we did stand counts, so we took stand, we took the number of plants damaged, we took the intensity of that damage, and we counted in any insect pests that we found. We did that early. It didn't amount to much this first year. We didn't have those damaging populations early. One thing looking at this year, and another reason why doing this multiple years is valuable, we didn't really have conditions that were favorable for early season insects at any of these sites. We planted that ore site earlier than I've ever planted any trial on a research farm. I think it was like April 10th or 13th. It was early. It was early for a university farm anyway. And it roared out of the ground and did fine, and it was warm while it was emerging, and we didn't have that problem. You know, none of these were really challenged during emergence during this first year. We challenge them during emergence, and we have an insect population there, yeah, then the story changes. And that's the value of doing this over time. Yeah, good question. Any other questions? See how much time I got to roll. Yeah, what's up?
So we went from extremely high green cloverworm in
Yep.
23 to almost absolutely nothing in 24. What's your thoughts there?
Yeah, that's a really interesting thought. The question is, what happened to green cloverworm? So in 2023, we had a pretty filthy outbreak of green cloverworm. We had it on our farm. We got good efficacy data on green cloverworm. 2024, we didn't hardly see them. And I don't know, going back to 2022, I think I saw 2 green cloverworms that whole summer. So we kind of had this boom and bust cycle. One thing to note about green cloverworm in particular, they're another one of these insects that is susceptible to just about every disease, every natural enemy. There's a lot of parasitism. There's a lot of fungal diseases that get them. That's actually the reason, if you look historically at green cloverworm recommendations, a lot of them are, there's actually papers written on the benefits of green cloverworm to the system as a biocontrol, like fodder, right? Like to get those pathogens and parasites and predators going. So they go through these boom and busts because of that. My suspicion is that 2023 was not particularly favorable for those pathogens, and that as a result of that, you build up the inoculum, just like you do with a plant disease. You build up that inoculum, then it's there, and then it's infecting them very early on in the season instead of later. But yeah, good point. And another excuse me, I don't know why I mumble so much. Another one of the reasons we want to repeat this year after year after year, we have these outbreak years. We have these green cloverworm years. We have these years where bean leaf beetles are big at the end of the season. We have years where Japanese beetle is big. We never know what it's going to be in a particular year. One of the most common questions I get asked this time of year is, what's the big insect going to be this coming season, right? And I laugh at it every time because I don't know. And, if anybody was going to know, hopefully it'd be the guy who freaking studies bugs for a living, right? There's not too many of those. There are not too many people who spend all their time thinking about bugs. I don't know. There's no way to know. It's all the variability in the weather, plus all the variability in ecology that goes along with that. When crops are planted, when the natural enemies are planted, that kind of thing. That's why we're doing this kind of work, not targeting any particular insect pest, but trying to look at the whole suite of pests. That's why we're doing this the way we're doing. Mm-hmm.
Along those lines then, with you getting more data points, is this a place that you could possibly use AI and get more information to find out the environmentals and everything that's in there that we could possibly predict in the future?
That is a Yeah, that is a great question. The question is about the potential to build a data set up and you use AI, and I would say other statistical tools as well, right, to kind of get around prediction, of what conditions favor these different insect outbreaks. Yeah, you're exactly right. We could do that, hopefully. We hope to do that with certainly the insect injury aspect of it, but there's a whole nother world of potential in this for looking at the effects of environmental conditions, the effects of soil type, the effects of nutrients. We're taking nutrient profiles at all these sites, we're taking soil samples, we're looking at everything we can measure. Over time, the value of that becomes really, really high. And you got to build up to that point. And part of my reason for wanting to do this study specifically, I wanted to do something that, God willing, we could carry on long term and continue to draw value out of long term. And it answers, or is attempting to answer, really some of the most common questions that I get that I often don't have an answer for, because if we do a trial like this next one, where we're targeting a particular insect, this last year it worked out. A lot of times it doesn't. We don't bat 1,000 putting out, whether it's chemical efficacy trials or any other sort of trial for insect control. There's just so much environmental variability, and that's part of what we're trying to reflect with this work. This study, I've done this a few times in the last few years. This is an efficacy study on bean leaf beetle. Okay, a couple of take-homes from this. One, we do not struggle to kill bean leaf beetle if we target with an insecticide in East Central Illinois. If you go down to the southern U.S., there's some areas where you get into some insecticide resistance with this species. We are not one of those areas. So if it's got bean leaf beetle on the label, you apply it while bean leaf beetle is in the field, you're probably going to kill them. You're probably going to kill them, and you're probably going to kill them pretty effectively. We did a nice job with that. One thing, that last slide there, if you can't read it, that's 14 days after application. What we're looking at there, okay, it's not like Warrior didn't make more bean leaf beetles, right? That's not what happened. What happened is, at that point, the residues for all these materials have gone away, and they're starting to move around this relatively small plot. In this case, we did our usual 10 by 40, a little more friendly for the guy in the backpack, which again was me for this. It makes it a lot less likely that I'm going to die when I do these trials. So by 14 days, we don't have residue anymore, right? In this case, we still didn't affect yield, and if you look at what those bean leaf beetle populations were, we're up to 150 in 25 sweeps. That means every time I swept that net through the field, I got 5 beetles. That's a pretty filthy population. Again, end of the season, when you get into that pod feeding kind of window, you get that defoliation late. Obviously, the defoliation, if it's happening at like R6, isn't going to amount to a whole lot. But that's what we're looking at there. And yeah, one of the take-home points of this, one thing when we talk about that R3 application in particular, our insect populations tend to be pretty low during that window. That's one of the things we see pretty consistently when we do this kind of work. The insecticides don't last very long. That's one of the reasons we recommend this scouting-based approach where we can. The other thing I wanted to point out, we've got this on-farm work with ISA. We'd love to have folks participate in it. Hopefully folks in this room, if this is something you're interested in evaluating on your farm, if you thought about a question that would be interesting to look at on your farm, let's do it. Let me know. If you can put the application out, we'll do the real fun of going out and counting these insects throughout the season. So you get some free scouting as a result of that as well. This other trial we're working on, also on farm. This is a tool Sean Conley up in Wisconsin developed along with his statistical guru, Spiros, where it's taken their data and it's going through an algorithm to sort of optimize things like row spacing, population, planting date, maturity group, different agronomic characteristics like that. Select a field, select a piece of ground, and choose what should be the optimal suite of those characteristics for profitability, right? That's what this tool is designed for. We're looking for folks to help us evaluate this on farm. Why is the entomologist doing this? You get roped into it, kind of. We're also evaluating along with this a scouting tool that guides our scouting efforts with NDVI collected via satellite. So the approach is we go out, we scout these fields thoroughly, and then we also scout them with the satellite assist. With the satellite assist, which gives us the red area in the field and says, here, go out, look at that area, see what you find in that area that from space looks like something's happening. That's what we're looking at the insect piece of that in particular. That part, from the on-farm standpoint, as a farmer, you don't have to do a damn thing. You just have to not point a shotgun at me when I walk to your field to do the scouting, okay? That's all you got to do for that. Just let us come out there, share some information with us. Obviously, the hard part of this is this trial evaluating these different combinations of agronomic characteristics. There's different combinations we can look at, so it's not that you have to change your row spacing, it's not that you have to change your variety. Depending on your situation, we can evaluate different characteristics here. If that's something you're interested in, please do reach out to me. And yeah, with that, I'm going to put up my contact information. I don't have long enough to make fun of Twitter, so I'm not going to make fun of Twitter. But if you try to get a hold of me on Twitter, be prepared to wait a while. The better way is to text me. Shoot me a text, and I'll get back to that pretty quickly. Shoot me a tweet, and I'll get back to it in an embarrassingly long time after that, and we'll, have that talk. But please don't hesitate to reach out if I can help with anything, and happy to answer any more questions if there's time and interest. Okay.
Any last questions?
Yeah, what's up?
Switching gears, will we have corn aphids? Will the aphids come back next year?
Oh my. Yeah, the question is if cornleaf aphid will come back next year. And I know you can guess what my answer is going to be. We don't know. We don't know if they're going to come back. And the thing about cornleaf aphid, most of what we see, there's a chance you get some overwintering in southern Illinois. No one's ever delineated that very effectively. You don't get overwintering here in Champaign. Almost everything we're seeing in Illinois in any given year with that species is coming in from the southwest. It's migratory. They fly in on storm systems. Okay, a lot of people last year associated it with beryl, and it did come in about that time, but we started getting calls the week before beryl. So it wasn't perfectly associated with that. But wind events, what these aphids do, they get up into the jet stream, and they ride that jet stream, and it drops them wherever. Folks call them aerial plankton, okay? They're not very good at directing their... They end up where the wind takes them, kind of. So to predict what's going to happen next year, it's something we just can't do. Another anecdote, I shared this with some folks at lunch earlier. So when you go back through and read these papers, you pick up these outbreaks. And it's not at regular intervals, okay? So it's not like I could say it's every 20 years, but sometimes it's 5 years, sometimes it's 20 years. But you'll read about like the big aphid outbreak of 1943 or 1957 or 1968. 86, I think, was the one we heard earlier this afternoon. Some places in the literature you see, oh yeah, it's these hot, dry conditions that cause that to happen. You see another paper reporting the exact same thing that said, it was cooler than normal, and therefore we got cornleaf aphids. It was wetter than normal, and therefore we got cornleaf aphids. There's two weather conditions you can associate with cornleaf aphid. One, if you have it and it's hot and dry, the injury that you see is going to be worse. It doesn't mean there's going to be more aphids, just that the impact they have is greater. Two, if you have a hard, drenching rain after that population is on there and established, it's going to go a long way towards knocking them off. And that's not just true of cornleaf aphid, but of aphids in general. That's the reason most of them ride on the underside of the leaves, is that a raindrop is, like 10 times the size of the aphid, so it is bad for them when those come down. But yeah, there's no ability to predict what conditions are going to lead to that. One, because it's never been established in the literature, and two, because it's not the conditions here. It's the conditions in maybe Texas, maybe Oklahoma, maybe Mississippi, depending on what way the wind's blowing and where the storm comes from. All right. You're sneaking up closer. I know I'm done now.
Yep.
I appreciate y'all's time. Thanks for having me, and don't hesitate to reach out if I can help you.